Hello friends, welcome back to my channel Biofric. So in this lecture video, I'll be starting with a topic called Cyclic Photophosphorylation. Okay, now what is the cyclic photophosphorylation? In the last class, I talked about the non-cyclic photophosphorylation that was following the Z scheme here, and what was getting formed is NADPH, right? So now in cyclic photophosphorylation, who is involved is solely PS1 system. PS1 system, also known as P700, absorbs the far red light of 700 nanometer and gets excited to P700 upstream. Okay, this is the excited state of the PS1. In cyclic photophosphorylation, PS2 is not at all involved, and since it's the cyclic flow, finally the electrons will move back, okay, to P700. And in cyclic photophosphorylation, solely just ATPs are formed, while in non-cyclic photophosphorylation we also saw that along with the NADPH, the proton gradient that was forming in the lumen was responsible for the formation of ATPs. But here only the ATPs will be formed in the cyclic photophosphorylation. At the same time, here you won't see any oxygen that will be evolved, since there is no oxygen evolving complex involved here. Okay, we are not at all concerned with the formation of oxygen in the case of cyclic photophosphorylation. So how does the relay of electrons happen in cyclic photophosphorylation? So as I said, once this particular P700 absorbs the light of wavelength 700 nanometer, around that it gets excited and it passes those electrons to AO. AO is again the modified chlorophyll, then to A1, which is phyloquinone, okay, also known as vitamin K1, and from phyloquinone it will move through iron sulfur proteins like X, A and B. Okay, all these three are the three different iron sulfur proteins that we saw in the cyclic or non-cyclic photophosphorylation as well. So from there on, the electron flows to ferredoxin to now cytochrome B6F complex. And since cytochrome B6F complex also was accepting the electrons from the quinone, somewhere the quinones are also involved here. Okay, so now from here again the plastocyanin, that was the copper containing compound, comes into play, and finally the electrons are passed on back to the PS1 system. So this is the cycle that keeps going. Now you might be wondering, if there is non-cyclic photophosphorylation, you're getting both of these molecules here that can be used in further dark reaction. What is the need of this particular cyclic photophosphorylation wherein no NADPH is formed? So for that matter, the answer is this cycle is favored when the system has enough amount of NADPH, but still there is a need of ATP to power other activities in the chloroplast. So you might be wondering, where is that ATP getting formed, right? So you remember in this complex, okay, the cytochrome B6F was pumping out protons. So from here, those protons will be pumped in the lumen. Okay, now since there'll be a proton gradient that will be formed in the lumen, you have your FO F1 pump and protons, and when they move out, ATP will be formed. So there is no production of NADPH, but there is production of ATP. So just remember, in cyclic photophosphorylation, since we are involving cytochrome B6F that was responsible for pumping in approximately 8 protons, okay, in the non-cyclic photophosphorylation, the same complex here is playing a major role, and all those protons that enter your lumen will be later pumped out back into the stroma. If you haven't seen the non-cyclic photophosphorylation video, I highly recommend you to go back and check that video out, and you will understand this even more, easily. So now your ATP is already formed. In addition to this concept, I would like you guys to understand another interesting concept. As I said, uh, we have PS1 and PS2. PS1 absorbs the light of 700 nanometer. It's of higher wavelength, and this one absorbs 680 nanometer. So in non-cyclic electron flow, the involvement, this is regarding the non-cyclic electron flow as well, okay, not directly related to the cyclic flow, but this concept is again very interesting. So stay tuned. Now in non-cyclic electron flow, what happens is that there is the involvement of two photosystems of two different they absorb the wavelength of two different two different wavelengths are getting absorbed by the systems, right? So if light energy available to PS1 and PS2 is not precisely matched. Like for example, the light energy in the environment here is just 700 nanometer, and there is no sufficient 680, or for that matter, let's say 680 is there, but it's not reaching 700 nanometer intensity. Then do you think this PS1 will work sufficiently? I don't think so. So for that matter, what is happening here is the plant is very smart here. It has its own different strategy. So in most efficient situations, the input energy would be the same to both photosystem. Most of the time it is the same, okay, the input energy. However, no single arrangement of pigment would satisfy this requirement because of the different times of the day, light intensity and spectral distribution tend to favor one photosystem over the other. So we know that in over a period in a day, if you go to see, the light intensity varies, the spectral distribution will later favor just one photosystem, like photosystem 2, or it will favor just one photosystem 2 or photosystem 1. So now how this problem is solved? Okay, so this problem can be solved by a mechanism that shifts the energy from one photosystem to the other in response to the different conditions. So as I said, I spoke about the chloroplast structure, and we have grana like this. Grana membrane has this thylakoid membrane, and in this thylakoid membrane we have all these photosystems and all, right? Sorry, membrane became membrane. Now this thylakoid membrane contains something called as protein kinases. Okay, it has something called as protein kinases. These are the kinases that will use ATP, okay, phosphorylation. And what do these protein kinases will do about this, shifting or making the photosystem light absorption efficient? So let's learn about that. So the protein kinases phosphorylate a specific threonine residue on LHC2. Now, where is this LHC2 situated? So just remember, PS2 has LHC2, while PS1 has LHC1. Okay, PS1 has LHC1, PS2 has LHC2. That's simple. Now, we are concerned about this LHC2 only. So, this LHC2 has a specific threonine residue. Okay, there is a threonine residue here that is actually phosphorylated. By whom? By this protein kinase. Okay, now phosphorylation begins the interaction of LHC2 with PS2. So now this LHC2 is basically interacting with the PS2, right? So as it gets phosphorylated by your protein kinase, this interaction between PS2 and LHC2 weakens, and it slowly will start, getting dissociated from PS2. Where it will move. So the LHC2 that is dissociating will move towards the stroma thylakoid. okay, out of the upraised region. Now if you remember, I spoke about, I don't know if I have mentioned this, but then there are two sides. Okay, this is your grana. Okay, grana. Now grana has upraised sides. This is your upraised sides, and this is your non upraised side. Now in the non-oppressed sites, your PS1 is prominent. Okay, PS1 is present more in amount in the non-oppressed side. That is not pressed. If you go to see in simple terms, what is this? So something that is pressed is oppressed. This is not exposed to the stroma basically. The grana thylakoid that is expressed to the stroma is non-oppressed. So non-oppressed has a high amount of PS1, while in the oppressed side the PS2 ratio PS1 is approximately 1.5:1. So you can see how the distribution varies. So basically in oppressed sites more amount of PS2 is present. Now from this PS2 wherein from this oppressed site your LHC will start dissociating, because previously obviously you can see that your PS2 will be LHC2 will be associated with PS2 from the oppressed side, so it will slowly start moving outwards near the grana, and when it is phosphorylated it will get associated with your PS1. Okay, now thus when LHC2 is not phosphorylated, it delivers more energy to PS2. Okay, so non phosphorylated will help its original partner, that is PS2. But when it gets phosphorylated, it will deliver more energy to whom? It will deliver more energy to PS1. So this protein kinase is activated when the plastoquinone, one of the electron carrier between your PS1 and PS2, accumulates in the reduced sites or reduced state. Okay, when it accumulates in the reduced state, obviously there'll be need. Okay, just remember now, when will your plastoquinone be reduced? When it becomes plastoquinol. I talked about the Q cycle as well, right? So when it gets most of them gets reduced, that means they are looking forward to move to the PS1 system, so that NADPH will be formed there later, right? So if the system senses that there is more amount of plastoquinol, this is quinol, because, or you can for simplicity you can say that it is plastohydroquinone. Okay, so there is no no problem, like you will make mistakes something like this, quinone or quinol. So you can say that plastohydroquinone also. When this gets accumulated more amount of this reduced form, obviously the protein kinase will sense that now it has to phosphorylate LHC2 so that it can move towards the PS1 system. PS1 will get more energy to get to reach that excited state, because it requires that 700 nanometers to reach that excited state, and that's how overall regulation is done, so that the energy is distributed to both the systems, because the problem here was two systems are absorbing the different wavelength, and over a period of time it has to be made sure that both the systems are working efficiently. And basically your protein kinase that phosphorylates the LHC2 on its threonine residue is doing so. So I hope you found both these topics that I taught today, that is, one is cyclic phosphorylation, and another one was regarding how to make the two photosystems efficiently work in terms of absorbing the light and getting excited and then transferring those electrons. I hope both these concepts were clear and were helpful. Thank you so much. See you in the next lecture.